Introduction: The Unseen Enemy. There is a war being fought at this very moment. It is a conflict older than human history, waged on a battlefield measured in micrometers, with trillions of casualties each year. It is a silent, invisible war fought not in the trenches of nations, but within the delicate tissues of every living person. Despite the gleaming towers of modern medicine, our powerful vaccines and arsenals of antibiotics, this ancient struggle continues. In many parts of the world, infectious diseases are not a historical footnote. They are a leading cause of death, a relentless siege that claims the most vulnerable among us, especially children. This is the story of that war. It is the chronicle of the invaders, the viruses, bacteria, fungi, and parasites, and of the intricate, violent, and often self-destructive defense of their target, the human body. Before the first shot is fired, we must understand the landscape. The human body is not a sterile sanctuary. It is a bustling, crowded ecosystem, home to a complex society of microbes known as the microbiome. Trillions of these commensal organisms live in a state of fragile truce with their host. They are the loyal citizenry, occupying territory, performing essential services, and defending the borders against foreign invaders simply by taking up space that a pathogen might otherwise claim. For the most part, this balance holds. But when the host's defenses are breached, when the government of the immune system is weakened, this delicate truce can shatter. Under the right conditions, even these oldest allies can turn against us, becoming opportunists, traitors, and killers. The war, you see, does not always begin with a foreign invasion. Sometimes, the most dangerous threat is the one that was inside you all along, waiting for a moment of weakness. Part I: The Rules of Engagement. Chapter 1: The Breach: Finding a Way In. Every invasion begins with a breach. The microbial world has spent millennia evolving sophisticated strategies to overcome the body's formidable defenses. Each surface, each passage, is a potential point of entry, and for every barrier there is an agent of infiltration designed to circumvent it. The skin is the first and most obvious line of defense, a multi-layered fortress wall of keratinase cells. Its surface is a harsh, inhospitable terrain, dry, acidic, and laced with antimicrobial fatty acids and defensins, chemical agents toxic to would-be invaders. For most microbes, it is impenetrable. But the fortress is not perfect. Siege engines in the form of mechanical injuries, a minor cut, a deep wound, a burn can tear down the walls. Trojan horses, like intravenous catheters or contaminated needles, can bypass the main gates entirely, delivering pathogens directly into the deeper tissues or the bloodstream. And then there are the special operations forces. Insect vectors, mosquitoes, ticks, fleas act as paratroopers, depositing their deadly cargo directly behind enemy lines. More insidiously, some invaders bring their own demolition tools. The larvae of the schistosoma parasite, for example, are like sappers. They don't need a pre-existing breach. They land on intact skin and release enzymes that dissolve the very mortar holding the cellular bricks together, melting a path for their entry. If an invader can bypass the skin, it may attempt to enter through the gastrointestinal tract, a treacherous, winding gauntlet filled with deadly traps. The first is the stomach, a churning river of acid so potent it can kill most organisms on contact. To demonstrate its power, consider the bacterium Vibrio cholerae. In clinical studies, neutralizing the stomach acid of healthy volunteers made them 10,000 times more susceptible to infection. Beyond the acid trap lies a viscous swamp of mucus, designed to ensnare pathogens. Pancreatic enzymes and bile detergents act as chemical weapons, destroying any microbe foolish enough to be encased in a lipid envelope. And patrolling this entire territory are the native inhabitants, the normal gut microbiota, acting as a relentless guerrilla force, competitively inhibiting any foreign colonizers. Yet, despite this fearsome defensive array, many pathogens have evolved countermeasures. The norovirus, the infamous scourge of cruise ships, is an armored transport, a non-enveloped virus resistant to acid, bile, and enzymes. The cysts of protozoa and the eggs of helminths are like armored personnel carriers, their tough outer coats allowing them to pass through the stomach's acid bath unharmed. Once they have survived the gauntlet, these invaders employ one of three primary attack strategies. Some, like Staphylococcus aureus in contaminated food, engage in pure chemical warfare. They don't need to invade the host at all. They simply leave behind a potent exotoxin, a pre-planted balm that causes acute food poisoning. Others, like V. cholerae, establish a beachhead, binding to the intestinal wall and launching a sustained artillery barrage of toxins that cause catastrophic fluid loss. And finally, there are the true infantry invaders like Shigella and Salmonella, which directly storm the mucosal walls, triggering a brutal form of urban warfare that results in ulceration, inflammation, and hemorrhage, the clinical state we call dysentery. The respiratory tract presents a different challenge, an airborne assault. With every breath, a blizzard of microscopic particles, dust, viruses, bacteria, fungi is drawn into the body. The airway is a high-traffic airspace protected by a sophisticated air defense system. Large particles are trapped by the mucociliary blanket, a sticky layer of mucus propelled constantly upward by millions of tiny cilia, like an escalator to the throat, where the invaders are swallowed and sent to their doom in the stomach. Smaller particles, those less than 5 microns in size, may evade this trap and reach the deep air sacs of the alveoli. There, they are met by the sentinels, resident alveolar macrophages that engulf and destroy them. But clever invaders have ways to defeat these defenses. The influenza virus uses a protein called hemagglutinin like a precision grappling hook, latching onto specific receptors on epithelial cells and tricking the cell into pulling it inside. Others are saboteurs. Bordetella pertussis, the agent of whooping cough, releases toxins that paralyze the cilia, shutting down the mucociliary escalator and allowing the bacteria to establish an infection. Perhaps the most cunning strategy belongs to Mycobacterium tuberculosis. It is the ultimate sleeper agent. It rides on the smallest dust particles into the deepest part of the lung, where it allows itself to be captured by a macrophage, only to deploy mechanisms that prevent its own destruction. It turns its prison into a sanctuary, a safe house where it can replicate, hidden from the rest of the immune system. Finally, the war can be a generational one. Through vertical transmission, the conflict is passed from mother to child. The invasion can occur across the placenta, during the passage through the birth canal, or even through breast milk. The timing of such an attack is critical. A rubella virus infection during the first trimester of pregnancy can cause devastating collateral damage, heart malformations, deafness, intellectual disability. The same virus, attacking in the third trimester, may have little effect. The battlefield, in this tragic theater of war, is the womb itself, and the stakes are the future of a developing life. Chapter 2, The Invasion Spreading the Infection. Establishing a beachhead is only the beginning. To succeed, an invader must expand its territory. Once inside, pathogens begin a strategic campaign of dissemination, moving through the body's intricate networks of highways and covert channels. Some secrete enzymes that act like chemical bulldozers, breaking down tissues to allow for slow, contiguous advance. More often, they exploit the body's own infrastructure. The lymphatic channels become microbial supply lines, allowing pathogens to travel from the initial site of infection to regional lymph nodes, which they can use as staging grounds for a wider assault. The bloodstream is the superhighway, the most efficient route of all, granting an invader access to every organ system in the body. And for some, the nerves themselves become secret passages. Viruses like rabies, poliovirus, and varicella zoster can infect peripheral nerves and begin a slow, terrifying infiltration along the axons, creeping inexorably toward the central nervous system, the body's command and control center. When a pathogen successfully enters the bloodstream and begins to spread, the conflict can escalate into a systemic crisis. This is sepsis. It is not merely an infection. It is the body's declaration of total war. It is a catastrophic system-wide panic response, a cytokine storm that unleashes fever, plummeting blood pressure, and rampant coagulation. This systemic inflammatory response can become more deadly than the invader that triggered it, leading to widespread organ failure and death, even in a previously healthy individual. The final act of any successful invasion is exfiltration. Microbes have evolved a remarkable variety of exit strategies to ensure their transmission to a new host, continuing their lineage. They can be shed from the skin, aerosolized in a cough or a sneeze, voided in urine or feces, or passed through the intimate contact of sexual transmission. Some are fragile, unable to survive long in the harsh outer world, and rely on rapid, direct person-to-person spread. Others are hardy like tuberculosis, which can linger in the air in tiny particles, traveling long distances to find its next victim. Some, like the helminths, release their eggs into the stool, where they hatch into larvae capable of penetrating the skin of the next unsuspecting host. Every strategy is a testament to a single, ruthless evolutionary imperative: survive, replicate, and spread. The war is never over, it simply moves to a new battlefield. Chapter 3: Cloak and Dagger, the Art of Evasion. The host is not a passive victim. It possesses a vast and complex military force, the immune system. This sets the stage for the most sophisticated theater of the war, a high-stakes game of espionage and counterintelligence. For every weapon the immune system deploys, the microbial world has engineered a countermeasure. This is the evolutionary arms race, and survival depends on the art of evasion. One of the most effective strategies is disguise. The immune system identifies enemies by recognizing specific molecules on their surface, known as antigens. In response, it generates antibodies that act like targeted homing missiles. But what if the enemy could change its appearance at will? This is the strategy of antigenic variation. Trypanosomes, the parasites that cause African sleeping sickness, are masters of this deception. They are covered in a dense coat of a single protein. Just as the immune system produces the perfect antibody to target this coat, a small number of the parasites switch to producing a completely different coat protein, rendering the host's entire arsenal obsolete. They are, in effect, spies who can change their face and uniform in an instant, always staying one step ahead of the security forces. The influenza virus takes this to another level. Through antigenic drift, it makes small mutations that alter its surface proteins. But through antigenic shift, two different viral strains can meet inside a single host cell and swap entire segments of their genetic code, creating a completely new, unrecognizable virus against which the population has no pre-existing immunity. This is how pandemics are born. If an invader cannot hide, it must fight. Phagocytes, like macrophages and neutrophils, are the frontline soldiers of the immune system, tasked with engulfing and destroying invaders. To counter them, many bacteria have developed body armor in the form of a thick, carbohydrate capsule. This slippery outer layer, found on pathogens like Streptococcus pneumoniae, makes it difficult for phagocytes to get a firm grip. Staphylococcus aureus employs a more cunning countermeasure, a surface molecule called protein A. It binds to antibodies, but it grabs them by the wrong end, the Fc portion, effectively disarming them so they cannot signal the phagocyte to attack. Other microbes are specialists in escaping capture. Listeria monocytogenes allows itself to be engulfed, only to use a pore-forming protein to punch its way out of the phagosome, becoming a fugitive in the vast city of the cytoplasm. Mycobacterium tuberculosis takes a different approach. It doesn't escape its prison. It sabotages the lock, producing proteins that prevent the phagosome from fusing with the lysosome, the cell's deadly digestive organelle. It remains safely inside its vesicle, turning a kill box into a nursery. The most advanced pathogens engage in high-level sabotage, targeting the very command and control systems of the immune response. T cells, the elite special forces, recognize infected cells by scanning for microbial fragments presented by MHC molecules. Several viruses, including herpesviruses and cytomegalovirus, have learned to jam this radar system. They produce proteins that bind to MHC molecules, preventing them from ever reaching the cell surface to sound the alarm. Some viruses release decoy receptors into the bloodstream, false signals that intercept the cytokines, the immune system's chemical messengers, before they can mobilize a response. A particularly insidious strategy is the exploitation of the body's own safety mechanisms. The PD-1 pathway is an immune checkpoint designed to prevent autoimmune reactions by shutting down overzealous T cells. In chronic viral infections like HIV and hepatitis C, the virus can manipulate this pathway, tricking the host's most powerful soldiers into a state of exhaustion, convincing them the fight is hopeless until they simply stand down. But perhaps the ultimate form of evasion is to disappear completely. This is the strategy of latency, the domain of the sleeper cell. Viruses like herpes simplex virus and varicella-zoster virus retreat into the quiet sanctuary of neurons. Epstein-Barr virus hides within B lymphocytes. In this latent state, they express few, if any, of their genes. They become silent, invisible, integrated into the very fabric of the host. They can lie dormant for years, even decades, evading all immune surveillance, waiting for the perfect moment, a weakening of the host's defenses to reactivate and strike from within, a ghost from a long-forgotten battle. Chapter 4: Weapons of Mass Destruction: How Invaders Cause Harm. Once an invader has breached the gates and evaded the sentinels, it must inflict damage. The microbial world has developed a terrifyingly diverse arsenal, from biological agents that hijack cellular machinery to chemical weapons that kill from a distance and trigger systemic chaos. The viral playbook is one of hostile takeover. A virus is the ultimate corporate raider. It brings no machinery of its own. It injects its genetic blueprint into a host cell and seizes control of the cell's replication factories, forcing them to churn out thousands of new viral copies. This process is often lethal. Some viruses, like poliovirus, actively sabotage the host by inactivating proteins essential for the cell's own functions. Others overwhelm the cell with such a massive burden of viral protein production that it triggers the cell's own self-destruct sequence, a process called apoptosis. Some viruses can even disable this self-destruct switch, keeping the cellular factory running for as long as possible. The most insidious outcome is transformation. Oncogenic viruses don't just kill the cell, they corrupt it, inserting their own genes into the host's DNA or inactivating key tumor suppressors, turning a loyal citizen of the body into a cancerous traitor that grows and divides without limit. The predilection of a virus for a certain cell type, its tropism, is often determined by the presence of specific receptors on the cell surface, a lock for which the virus has evolved the key. This is why HIV targets T cells via the CD4 receptor, and Epstein-Barr virus targets B cells via the CD21 receptor. The bacterial arsenal is, if anything, even more varied. The blueprints for their most potent weapons are often found on mobile genetic elements, plasmids, and bacteriophages that can be passed between bacteria like illicit arms deals, spreading genes for toxins and antibiotic resistance. Many bacteria have learned to coordinate their attack through a process called quorum sensing. They secrete small molecules, and only when the population density is high enough, when the concentration of these molecules reaches a critical threshold, do they launch their assault, switching on virulence genes en masse to overwhelm host defenses in a sudden, synchronized strike. Many also build fortifications. Biofilms are slimy, viscous fortresses of extracellular polysaccharides, enemy encampments built on surfaces like intravascular catheters or artificial joints. Within these strongholds, bacteria are shielded from antibiotics and immune cells, allowing them to persist and cause chronic, relapsing infections. The weapons themselves are marvels of malevolent engineering. They fall into three main classes. First, there is endotoxin. This is not a secreted weapon but an integral part of the outer membrane of gram-negative bacteria, a molecule called lipopolysaccharide, or LPS. It is a booby trap. As long as the bacterium is alive, it is relatively harmless. But when the bacterium is killed by the immune system, the LPS is released, and it is a potent trigger for a massive, chaotic, and often fatal immune overreaction. High levels of endotoxin can induce septic shock, disseminated intravascular coagulation, and respiratory failure. The weapon's true power lies in its ability to turn the host's own defensive strength into a force of self-destruction. Second, there are the exotoxins. These are secreted proteins, precision-guided munitions designed for specific tasks. Many take the form of B toxins. The B subunit is the targeting system, binding to a specific receptor on a host cell. The A subunit is the warhead, which is delivered into the cell's cytoplasm to carry out its mission. The toxins of cholera and diphtheria work this way, disrupting cellular signaling or halting protein synthesis. The neurotoxins produced by Clostridium botulinum and Clostridium tetani are among the most potent poisons known. They don't kill neurons, they simply cut the communication lines. Botulinum toxin blocks the release of acetylcholine at the neuromuscular junction, causing a flaccid paralysis. Tetanus toxin blocks the release of inhibitory neurotransmitters, leading to violent, uncontrollable muscle spasms. Finally, there are the superantigens. This is the weapon of mass chaos. Normally, T-cell activation is a highly specific process, with a single T-cell recognizing a single antigen. Superantigens, produced by bacteria like Staphylococcus aureus, bypass this entire system of checks and balances. They act as a master key, binding to conserved regions of T-cell receptors and indiscriminately activating up to 20% of the body's entire T-cell population at once. The result is a cytokine tsunami, a flood of inflammatory mediators that plunges the body into the systemic meltdown of toxic shock syndrome. Chapter 5. Friendly fire when defenses turn destructive. There is a tragic irony at the heart of pathology. In the brutal war against infection, the most devastating damage is often not inflicted by the invader, but by the host's own defenders. The immune system is a powerful but blunt instrument, and its victories are never clean. The very act of fighting the inflammation, the release of cytotoxic chemicals, the targeted killing of infected cells inevitably causes collateral damage. In many diseases, the pathology is not the story of what the microbe did to the host, but what the host did to itself in its desperate attempt to survive. Consider the battle against Mycobacterium tuberculosis. The immune system's brilliant strategy is to build a wall around the unkillable foe, forming a structure called a granuloma. This siege is often successful at containing the infection, but the process itself is destructive. At the heart of the granuloma, tissue dies in a process of caseous necrosis, and over time, the entire structure is replaced by a fibrotic scar. The lung is permanently damaged. The very mechanism of winning the local battle contributes to losing the larger war for organ function. The scar of a past victory becomes a future liability. In chronic hepatitis B or C infection, it is not the virus itself that destroys the liver. It is the host's own cytotoxic T cells, which in their relentless campaign to eliminate infected hepatocytes, gradually demolish the liver's architecture, leading to cirrhosis. Sometimes the immune system's targeting systems go awry. In rheumatic heart disease, antibodies produced against a streptococcal protein cross-react with proteins in the heart, leading the immune system to launch a devastating autoimmune attack on its own cardiac valves. In pathology, the story of an infection is often read in the scars left behind by these battles. By examining the tissue under a microscope, we can identify the signature of the conflict, a distinct pattern of damage that tells us about the nature of the invader and the type of war that was fought. Let us examine the evidence. This is a damage assessment report from the front lines. There are 5 major patterns of tissue reaction, 5 signatures of war. First, suppurative inflammation. Here, we see the aftermath of a pyogenic assault. The battlefield is flooded with neutrophils, the infantry of the immune system. It is a scene of liquefactive necrosis, a churning sea of dead and dying cells we call pus. This is the signature of a swift, brutal, frontal assault by extracellular bacteria like Staphylococcus or Streptococcus. The destruction can be localized, as in an abscess, or it can consume an entire lobe of the lung. Second, mononuclear and granulomatous inflammation. A different kind of war. This is a chronic siege, a protracted conflict against an intracellular enemy that cannot be easily eradicated. The defenders here are the specialists, macrophages and lymphocytes. In response to persistent agents like Mycobacterium tuberculosis or the eggs of a schistosome worm, they form granulomatous living walls of activated macrophages, attempting to contain the unkillable foe. But the siege takes its toll, leaving behind the caseous, necrotic ruins of a long and costly struggle. Third, the cytopathic cytoproliferative reaction. This is not a battle, it is a hostile takeover. It is the signature of a viral agent. We see cell necrosis, yes, but also bizarre, uncontrolled proliferation. Some viruses force infected cells to fuse into giant, monstrous multinucleated syncytia. Others, like human papillomavirus, push the cell into cancerous growth. The virus isn't just killing, it is rewriting the very nature of the cell, turning it into something alien. Fourth, tissue necrosis. Sheer, unadulterated destruction, often with a shocking absence of inflammatory cells. This is the signature of a powerful, toxin-wielding agent like Clostridium perfringens, the cause of gas gangrene. There is no grand battle here, no legions of neutrophils rushing to the scene. There is only the silent, rapid death of tissue. It is the chilling aftermath of a chemical weapon attack, where the damage is so swift and severe that the body has no time to mount a defense. And finally, chronic inflammation and scarring. This is the long-term consequence of any protracted war. The scar. In chronic hepatitis, we see the liver's elegant architecture shattered and replaced by dense, functionless bands of fibrous tissue. In schistosomiasis, the liver develops a pipe stem fibrosis around the tract eggs of the parasite. This is the price of survival, a landscape permanently and irrevocably altered by the memory of infection. These patterns rarely appear in pure form. A single battlefield may show signs of multiple conflicts, a complex and overlapping history of damage. But by reading these signatures, we begin to understand the fundamental rules of engagement in the war within. Part Roman II, Case Files from the Front Lines. Case Files, the viral insurgency. Viruses are the ultimate intracellular parasites. They are minimalist agents of chaos, little more than a genetic blueprint wrapped in a protein coat. They cannot replicate on their own. They must hijack the machinery of a living cell to do their bidding. Their strategies range from swift, transient assaults to lifelong, latent occupations. The blitzkrieg acute infections. Some viruses specialize in a rapid, hit-and-run strategy. They cause acute, transient infections that are ultimately cleared by an effective immune response, but not before they have caused their signature damage and spread to a new host. Case file: measles. The measles virus is a master of efficient transmission, an airborne agent of the paramyxoviridae family that spreads via respiratory droplets. Its initial target is the respiratory tract, but from this beachhead, it launches a systemic invasion. Using a specific receptor called SLAM-phone, it infects the sentinels of the immune system itself, lymphocytes, dendritic cells, and monocytes, and spreads through the lymphatic system into the blood. This viremia carries it to the skin, the conjunctiva, the urinary tract, and the central nervous system. The characteristic reddish-brown rash is not caused by the virus directly, but is a sign of friendly fire, the T-cell-mediated immune response attacking infected cells in the small blood vessels of the skin. In the lymphoid tissues, the virus leaves a pathognomonic signature, massive, multinucleated giant cells, known as Werthen-Finkeldy cells, visible under the microscope like the footprints of a giant. The virus's most dangerous weapon, however, is its ability to cause a profound, transient immunosuppression, disabling the host's defenses and opening the door for deadly secondary bacterial infections. This is why a simple childhood disease can become a killer, especially in malnourished populations. Case file: mumps. A cousin of measles, the mumps virus also enters through the respiratory tract and spreads through the blood. Its primary target, however, is glandular tissue, most famously the parotid salivary glands. Infection of the ductal epithelial cells leads to inflammation, edema, and the classic painful swelling. But the virus does not always stop there. It can spread to cause a painful orchitis in the testes, which, due to the tight, unyielding capsule of the testes, can lead to swelling so severe that it compromises the blood supply, causing infarction and potential sterility. It can also cause pancreatitis and, in up to 15% of cases, aseptic meningitis, making it a systemic threat far more serious than its reputation suggests. Case file: poliomyelitis. The poliovirus is an enterovirus, a small, unencapsulated RNA virus transmitted by the fecal-oral route. It is a gut pathogen that replicates in the lymphoid tissue of the pharynx and ileum before spreading to the blood. In over 99% of cases, the infection stops there, controlled by a robust antibody response. But in a fateful few, the virus executes its most devastating maneuver: it invades the central nervous system. It may travel through the blood or, more terrifyingly, creep backward along the axons of motor neurons. Its target is specific and catastrophic: the motor neurons of the spinal cord and brainstem. By destroying these cells, the virus severs the connection between the brain and the muscles, resulting in irreversible flaccid paralysis. A mild intestinal infection becomes a lifelong sentence of disability, and, if the muscles of respiration are affected, a death sentence. The long war, latent and chronic infections. Not all viruses are content with a swift attack. The most insidious are those that have learned to play the long game, establishing a permanent presence within the host. These are the herpesviruses, a family of large, double-stranded DNA viruses that are masters of latency. They cause an acute primary infection, but then retreat into a dormant state, hiding from the immune system for a lifetime, ready to reactivate when defenses are low. Case file: herpes simplex virus (HSV). There are two closely related types, HSV-1 and HSV-2. They replicate in the skin and mucous membranes, causing the characteristic vesicular lesions, cold sores, or genital herpes. But this is just a distraction. Their true objective is to invade the sensory neurons that innervate the site of infection. They travel up the axons to the neuronal cell body in the sensory ganglia, and there, they go dark. The viral DNA persists in the nucleus of the neuron, a silent, sleeping passenger. Only a few non-coding RNA transcripts are made, perhaps to maintain this dormant state and prevent the host cell from undergoing apoptosis. Reactivation, often triggered by stress or immunosuppression, sends new virus particles hurtling back down the axon to the original site, causing a recurrent lesion. HSV has also evolved potent immune evasion tactics, producing proteins that inhibit MHC class I presentation, effectively hiding infected cells from cytotoxic T cells, and decoy receptors that neutralize antibodies and complement. While usually a localized nuisance, HSV-1 can cause a devastating, fatal sporadic encephalitis, and infection with HSV-2 is known to dramatically increase the risk of acquiring and transmitting HIV. Under the microscope, HSV leaves a distinct calling card: large, pinkish intranuclear inclusions called Cowdrey type of bodies and the fusion of infected cells into multinucleated syncytia. Case file: varicella zoster virus VZV. VZV plays by the same rules as HSV but on a grander scale. The primary infection is chickenpox, a systemic disease transmitted by respiratory aerosols that causes a widespread vesicular rash. Like HSV, the virus then retreats into sensory ganglia, establishing a lifelong latent infection. Decades later, a decline in immunity can allow the virus to reactivate. But instead of a localized recurrence, the virus erupts along the entire length of a single sensory nerve, causing shingles or herpes zoster. It infects the skin of a single dermatome, producing the unilateral band of intensely painful vesicles. The pain is a direct result of the concomitant inflammation of the nerve root, a radiculoneuritis that can persist for months or years as postherpetic neuralgia. Case file: cytomegalovirus CMV. CMV, is the great opportunist of the herpesvirus family. In healthy individuals, it typically causes an asymptomatic or mild mononucleosis-like illness before establishing latency in monocytes and their bone marrow precursors. But in the immunocompromised transplant recipients, AIDS patient CMV is a devastating systemic pathogen. Reactivation leads to widespread dissemination, causing life-threatening pneumonitis in the lungs and a destructive colitis in the gut. Its name, cytomegalovirus, describes its signature effect on infected cells. It causes them to swell to a gigantic size, their nuclei dominated by a massive basophilic inclusion surrounded by a clear halo, the pathognomonic owls, eye inclusion. Congenital infection, acquired in utero from a mother with a primary infection, can be catastrophic, causing intrauterine growth restriction, jaundice, and severe brain damage, including microcephaly and calcifications, leaving surviving infants with permanent neurologic deficits. Case file, Epstein-Barr virus EBV. EV, another herpesvirus, is the agent of infectious mononucleosis. Transmitted through saliva, it targets B lymphocytes, using the CD21 receptor to gain entry. In some B cells, it replicates and lyses the cell. In most, however, it establishes a latent infection, transforming the B cell into an immortalized, proliferating cell factory. This polyclonal B cell proliferation is what drives the disease. The host mounts a powerful counterattack, primarily with cytotoxic T lymphocytes (CTLS). The characteristic atypical lymphocytes seen in the blood of a mononucleosis patient are not the infected B cells, they are the army of activated CTLs rising up to fight them. This massive T cell response is what causes the sore throat, the generalized lymphadenopathy, and the splenomegaly so profound it puts the spleen at risk of rupture. ABV is also a transforming virus implicated in the pathogenesis of several human cancers, including Burkitt lymphoma and nasopharyngeal carcinoma, a stark reminder that the line between infection and malignancy can be perilously thin. Case files, the bacterial onslaught. Bacteria are the prokaryotic workhorses of the microbial world. They are single-celled organisms, far more complex than viruses, capable of independent life and reproduction. Their diversity is staggering, and their capacity to cause disease is protean. We classify them broadly by the composition of their cell wall, using a 19th century staining technique developed by Hans Christian Gram. The gram-positives, with their thick peptidoglycan wall, and the gram-negatives, with their thin wall and outer lipopolysaccharide membrane, represent two fundamentally different architectures for waging war. The gram-positive menace. These organisms are defined by their thick cell walls that retain the crystal violet stain in the Gram procedure. They are responsible for some of the most common and destructive suppurative infections known to medicine. Case file, Staphylococcus. Staphylococcus aureus is a pyogenic gram-positive coccus that arranges itself in clusters resembling bunches of grapes. It is a versatile and ruthless pathogen, responsible for a staggering array of diseases, skin lesions like boils and carbuncles, deep abscesses, bone infections, osteomyelitis, pneumonia, heart valve infections, endocarditis, and systemic syndromes like toxic shock syndrome. Its virulence is due to a vast arsenal of weapons. It expresses surface proteins that allow it to bind to host molecules like fibrinogen and fibronectin, essentially cloaking itself in host material to adhere to tissues. Its polysaccharide capsule helps it stick to artificial surfaces like catheters and prosthetic valves, while also resisting phagocytosis. Its protein binds antibodies backward, neutralizing them. It secretes a host of membrane-damaging toxins that lyse red blood cells and phagocytes. Its exfoliative toxins are serine proteases that specifically cleave desmoglein-1, the protein that glues epidermal cells together, causing the skin to peel away in sheets in staphylococcal scalded skin syndrome. And its superantigens are the weapons of mass chaos that trigger toxic shock syndrome. The rise of methicillin-resistant S. aureus MRSA, first in hospitals and now in the community, has made this ancient foe one of the most pressing threats of the modern antibiotic era. Case file, Streptococci and Enterococci. Streptococci, gram-positive cocci that grow in chains, are another family of formidable pyogenic pathogens. Streptococcus pyogenes group. A strep is infamous for causing strep throat, but also scarlet fever, the skin infection erysipelas, and the terrifyingly rapid flesh-eating necrotizing fasciitis. Its virulence is enhanced by a capsule and a surface M protein that both inhibit phagocytosis. But its true danger lies in the aftermath of infection. The immune response it provokes can lead to devastating post-infectious syndromes. Antibodies against its M protein can cross-react with cardiac proteins, causing the autoimmune heart damage of rheumatic fever. Immune complexes of streptococcal antigens and host antibodies can deposit in the kidneys, causing post-streptococcal glomerulonephritis. Streptococcus pneumoniae, with its critical anti-phagocytic capsule, is a leading cause of community-acquired pneumonia and bacterial meningitis. Enterococci, once considered low-virulence gut commensals, have emerged as major hospital-acquired pathogens, not because of exceptional virulence, but because of their extraordinary resistance to antibiotics. Case file: diphtheria. Caused by Corynebacterium diphtheriae, this disease is a testament to the power of a single devastating toxin. The bacterium itself remains localized to the upper respiratory tract, where it causes epithelial necrosis and induces a dense fibrinosuppurative exudate. This coagulates into a tough, dirty gray pseudomembrane that can cover the tonsils, pharynx, and larynx, potentially obstructing the airway and causing asphyxiation. But the real damage is done by the phage-encoded B diphtheria toxin that enters the bloodstream. The asubunit is an enzyme that inactivates elongation factor -2 EF-2, a protein essential for protein synthesis in host cells. A single molecule of toxin can inactivate over 1 million EF-2 molecules, shutting down cellular life and causing systemic damage to the heart, nerves, and other organs. Immunization with a toxoid, an inactivated form of the toxin, is one of public health's greatest triumphs, rendering this once common killer a rarity in vaccinated populations. Case file: anthrax. The agent is Bacillus anthracis, a large, spore-forming gram-positive rod. Its spores can survive for decades in the soil, making it a persistent threat to livestock and, by extension, humans. Anthrax presents in 3 forms. Cutaneous anthrax, the most common, begins as a skin papule that evolves into a vesicle and then a necrotic ulcer covered by a characteristic black eschar. Gastrointestinal anthrax, from eating contaminated meat, is rare but often fatal. The most feared form is inhalational anthrax, the agent of bioterrorism. Inhaled spores are carried to mediastinal lymph nodes where they germinate. The bacteria then release their potent toxins, causing a hemorrhagic mediastinitis that rapidly progresses to shock and death. The anthrax toxins are a sophisticated 3-part system. The protective antigen PA is the B subunit, which binds to host cells and forms a pore. Through this pore, it delivers one of 2 subunits, edema factor AF, an adenylate cyclase that disrupts cell function, or lethal factor LF, a protease that destroys key signaling kinases, leading to cell death. It is a molecular syringe of remarkable precision and lethality. The gram-negative offensive. Gram-negative bacteria, with their thin cell wall and protective outer membrane rich in endotoxin, present a different set of challenges. They are a diverse group, responsible for everything from meningitis to plague. Case file: Neisseria. These gram-negative diplococci, shaped like coffee beans, include 2 major pathogens. Neisseria meningitidis is a leading cause of bacterial meningitis, especially in adolescents and young adults living in close quarters like college dorms or military barracks. It spreads via the respiratory route, and its polysaccharide capsule is its key defense, protecting it from complement-mediated killing in the bloodstream. Individuals with inherited defects in the late components of the complement system C5-C9 are exquisitely susceptible to severe, disseminated infection. Neisseria gonorrhoeae, the agent of gonorrhea, is a major sexually transmitted infection. It uses long pili and outer membrane OPA proteins to adhere to and invade epithelial cells of the urogenital tract. A key to its success is its mastery of antigenic variation. Through complex genetic recombination, it can constantly change the proteins that make up its pili and OPA adhesins, allowing it to evade the host antibody response and cause recurrent infections. Case file: pertussis whooping cough, caused by the Coccobacillus bordetella pertussis. This highly communicable disease is characterized by paroxysms of violent coughing. The bacterium colonizes the bronchial epithelium, using a filamentous hemagglutinin to bind tightly to cilia. From there, it unleashes a suite of toxins. Its signature weapon is the pertussis toxin, and a B toxin that, like cholera toxin, ADP ribocylates G proteins, disrupting host cell signaling and impairing the recruitment and function of neutrophils and macrophages. It effectively disarms the local immune response and paralyzes the cilia, allowing a thick, mucopurulent exudate to build up, triggering the characteristic cough. Case file: plague. The agent of the Black Death, Yersinia pestis, is a gram-negative bacterium transmitted from rodents to humans by flea bites. Once injected into the skin, the bacteria travel to regional lymph nodes, causing them to swell into massive, hemorrhagic, necrotic masses called buboes, the hallmark of bubonic plague. If the infection reaches the lungs, it causes a necrotizing pneumonia and can be transmitted from person to person via aerosols, the terrifying pneumonic plague. Its virulence is encoded on a plasmid, which contains the genes for a type Roman 3 secretion system. This is a molecular syringe that injects a cocktail of bacterial proteins, called YAPs, directly into host immune cells. These YAPs are sabotage agents of the highest order. Some paralyze the cell's cytoskeleton to block phagocytosis, while others inhibit signaling pathways to shut down the production of inflammatory cytokines. The bacterium effectively neutralizes the immune response before it can even begin, allowing for massive, unchecked proliferation. The intracellular specialists. Some bacteria have taken evolution to its logical extreme, abandoning independent life altogether. These are the obligate intracellular bacteria, organisms that can only replicate within the protected confines of a host cell. Case file: chlamydia. Chlamydia trachomatis is the most common bacterial sexually transmitted infection in the world. As an obligate intracellular pathogen, it has a unique two-stage life cycle. The infectious form is a small, metabolically inert elementary body. It uses a type Roman 3 secretion system to inject proteins into a host cell, inducing its own uptake. Once inside, it transforms into a larger, metabolically active reticulate body, which replicates within a protected membrane-bound vacuole, stealing ATP and amino acids from the host cell. Eventually, the reticulate bodies condense back into elementary bodies, which are then released to infect new cells. This stealthy life cycle allows it to cause often asymptomatic infections in the urogenital tract that, if left untreated, can lead to chronic inflammation, scarring, and infertility, particularly in women. Certain serotypes cause the more aggressive disease lymphogranuloma venereum, characterized by granulomatous inflammation and stellate abscesses in the lymph nodes. Case file: rickettsia. The rickettsia are another group of vector-borne, obligate intracellular bacteria. They are the agents of typhus and spotted fevers, like Rocky Mountain spotted fever. Their primary target is the vascular endothelium. After being injected by a tick or louse bite, they enter endothelial cells, escape into the cytoplasm, and proliferate. This widespread infection of the body's blood vessel lining causes endothelial dysfunction and injury, leading to increased vascular permeability, edema, hemorrhage, and thrombosis. The characteristic petechial rash of these diseases is a direct visualization of this vascular damage. In severe cases, this widespread vasculitis can lead to shock, organ failure, and death. Case files: the fungal takeover. Fungi are eukaryotes like us. They exist as single-celled yeasts or filamentous molds. Most are harmless environmental organisms, but a select few have adapted to become formidable pathogens, particularly in hosts whose immune defenses are compromised. Case file: Candidiasis. Candida albicans is the most common fungal pathogen of humans. It is a normal commensal, a peaceful resident of our skin, mouth, gut, and vagina. In a healthy host, it is kept in check by the immune system and competing bacteria. But when the balance is disturbed by antibiotics, immunosuppression, or diabetes, Candida becomes an opportunistic invader. It can cause superficial infections like oral thrush, a white pseudomembrane of matted fungi and inflammatory debris, or vaginitis. In severely immunocompromised patients, particularly those with neutropenia, it can invade the bloodstream and disseminate throughout the body, causing life-threatening systemic disease with microabscesses in the kidneys, heart, brain, and liver. A key to its virulence is its ability to switch between a budding yeast form and an invasive filamentous form with pseudohyphae. It also forms biofilms on medical devices, making it highly resistant to antifungal drugs. Case file: Cryptococcosis. Cryptococcus neoformans is an encapsulated yeast found in soil and bird droppings. Inhaled into the lungs, it can cause a mild, often asymptomatic primary infection. Its true danger lies in its profound neurotropism. In immunocompromised hosts, particularly AIDS patients, the fungus can spread from the lungs to the central nervous system, causing a devastating meningoencephalitis. Its most potent weapon is its thick polysaccharide capsule, a gelatinous shield that inhibits phagocytosis and cripples the immune response. In the brain of a severely immunocompromised patient, the yeast can proliferate with almost no inflammatory reaction, filling the perivascular spaces and creating characteristic soap bubble lesions visible on imaging. Case file: Aspergillosis. Aspergillus is a ubiquitous mold. We inhale its airborne spores every day. In a healthy person, alveolar macrophages and neutrophils efficiently clear them. But in the immunocompromised, especially neutropenic patients, or those on high-dose corticosteroids, Aspergillus is a deadly invasive pathogen. The spores germinate in the lung, forming hyphae that invade tissues. A hallmark of invasive aspergillosis is its tendency to invade blood vessels, causing thrombosis, hemorrhage, and infarction. This leads to the formation of characteristic target-like lesions in the lung, rounded areas of necrosis with a hemorrhagic border. Under the microscope, Aspergillus is identified by its septate hyphae that branch at acute, 40-degree angles. Case file: Mucormycosis. The fungi of the order Mucorales are another group of environmental molds that cause opportunistic infections, particularly in diabetics with ketoacidosis and neutropenic patients. They are even more aggressive than Aspergillus. The most common presentation is rhinocerebral mucormycosis, where the infection begins in the nasal sinuses and spreads with terrifying speed to the orbit and brain, causing massive tissue necrosis. Like Aspergillus, they are angoinvasive, but their hyphae are morphologically distinct. They are nonseptate and branch at wide, often 90-degree angles. The infection is an absolute medical emergency, often requiring aggressive surgical debridement in addition to antifungal therapy. Case files: The parasitic infiltrators. Parasites represent the pinnacle of complex host-invader relationships. From single-celled protozoa to multicellular helminth worms, these organisms have co-evolved with their hosts for millennia, developing intricate life cycles and sophisticated mechanisms of survival. The protozoan threat. Case file: Malaria. Malaria, caused by the protozoan Plasmodium, is one of humanity's greatest scourges. Transmitted by the bite of the Anopheles mosquito, the parasite begins its life cycle with a silent phase in the liver. From there, thousands of merozoites are released to begin the erythrocytic stage, the invasion of red blood cells. Inside the red cell, the parasite consumes hemoglobin, growing and dividing until the cell ruptures, releasing a new wave of merozoites to infect more cells. It is this synchronized rupture of red cells that causes the classic paroxysms of fever and chills, a storm triggered by the release of parasitic material in host cytokines. The species Plasmodium falciparum is by far the most deadly. It can infect red cells of any age, leading to dangerously high levels of parasitemia. More critically, it remodels the surface of the infected red cell, causing it to become sticky. These altered cells adhere to the endothelium of small blood vessels, a process called sequestration, particularly in the brain. This blocks blood flow, causing the ischemia and hypoxia that lead to cerebral malaria, coma, and death. P. falciparum also employs antigenic variation, constantly changing the adhesion proteins on the red cell surface to evade the antibody response. The immense selective pressure exerted by malaria on the human genome is evident in the high prevalence of genetic traits like sickle cell anemia and thalassemia, in endemic regions deadly diseases in their own right, but which offer a survival advantage against this even deadlier parasite. Case file: Leishmanioses. Transmitted by the bite of a sandfly, Leishmania is an intracellular parasite that targets macrophages. The infectious promastigotes are injected into the skin, where they are eagerly phagocytosed by macrophages. But this is a trap. The parasite manipulates the complement system to facilitate its own uptake, then transforms into an amastigote form within the phagolysosome, where it not only survives but thrives, replicating until it ruptures the cell. The outcome of the infection is determined by the host's T cell response. A strong Thone response with production of interferon gamma activates macrophages to kill the parasites, resulting in a localized, self-healing skin ulcer cutaneous leishmaniasis. A weak Thone response or a dominant Th4 response allows the parasite to spread unchecked. In visceral leishmaniasis or Kala-Azar, the parasites disseminate throughout the mononuclear phagocyte system, causing massive hepatosplenomegaly, fever, pancytopenia, and profound wasting. It is a disease whose clinical spectrum is a direct reflection of the host's immunological civil war. Case file: Chagas disease. American trypanosomiosis, caused by Trypanosoma cruzi, is transmitted by the feces of triatomine-kissing bugs. The parasite can invade nearly any cell type, but has a devastating predilection for cardiac muscle and the nerve cells of the myenteric plexus in the gut. The acute phase is often mild. The chronic phase, which develops years later, is where the true damage occurs. Persistent, low-level infection triggers a chronic inflammatory response in the heart, leading to a progressive, dilated cardiomyopathy that is a major cause of sudden cardiac death in endemic areas of Latin America. The parasite may also induce an autoimmune response, where T cells and antibodies targeting the parasite cross-react with host tissues, amplifying the damage. Destruction of the myenteric plexus leads to the grotesque dilation of the esophagus and colon, known as megaesophagus and megacolon. The metazoan invasion helminths. Helminths, or parasitic worms, are multicellular organisms that have adapted to live within a host. Their infections are often chronic, and the pathology is typically caused not by the adult worms themselves, but by the host's intense inflammatory response to their eggs or migrating larvae. Case file: Schistosomiasis. The life cycle of this fluke worm is a marvel of adaptation. Infectious cercariae larvae in fresh water penetrate human skin and migrate through the circulation to the liver, where they mature into adult worms. The male and female worms then pair up and migrate to their final home in the mesenteric or pelvic veins. There, the female lays hundreds or thousands of eggs per day. The eggs are the true weapons of the disease. They must pass through the wall of the intestine or bladder to be excreted and continue the life cycle. To do this, they secrete enzymes that digest tissue, but they also elicit a powerful host granulomatous inflammatory response that helps to push them through the wall. It is the eggs that do not escape, those that are swept by the portal circulation into the liver, that cause the major pathology. The host forms massive granulomas around these trapped eggs, and over years, this leads to a unique and devastating pipe stem fibrosis of the liver. This fibrosis obstructs portal blood flow, causing portal hypertension, massive splenomegaly, and bleeding from esophageal varices. It is a disease where the pathology is almost entirely due to friendly fire. Case file: Lymphatic filariasis. Caused by the nematodes Wuchereria bancrofti and Brugia malayi, this disease is transmitted by mosquitoes. The adult worms live exclusively in the lymphatic channels and lymph nodes. Their presence and the host's inflammatory response to them causes obstruction of lymphatic flow. Over years of chronic infection, this leads to a persistent, disfiguring lymphedema of the limbs and genitalia. In its most extreme form, the skin becomes thickened and hyperkeratotic, a condition known as elephantiasis. The worms themselves are masters of immune modulation, secreting a host of molecules that dampen the immune response and allow them to survive for years. Much of the inflammation is now thought to be triggered by symbiotic Wolbachia bacteria that live within the worms. When the worms die, the release of these bacteria provokes a powerful host reaction. Conclusion: the evolving war. The war is not static. The battlefield is constantly changing, shaped by human behavior, environmental disruption, and the relentless engine of microbial evolution. In the last half-century, we have witnessed the emergence of entirely new threats and the re-emergence of old foes in new, more dangerous forms. Human demographics and technology have opened new fronts in the war. The global travel network that connects our world in hours also provides a superhighway for pathogens, allowing an outbreak in a remote village to become a global pandemic in weeks, as the world learned with SARS-CoV-2. Environmental changes, like the reforestation of the northeastern United States, have led to an explosion in the deer and tick populations, driving the epidemic of Lyme disease. The widespread, often indiscriminate use of antibiotics has exerted a massive selective pressure on the bacterial world, leading to the terrifying rise of multi-drug-resistant organisms, superbugs like MRSA and carbapenem-resistant Klebsiella, for which our medical arsenal is dwindling. We have also faced entirely novel agents, pathogens that have made the leap from animal reservoirs into the human population. HIV emerged from primates to cause the AIDS pandemic. Ebola, Nipah virus, and SARS coronaviruses have emerged from bats, causing explosive outbreaks with high mortality. Zika virus, long a minor pathogen, re-emerged with a terrifying new capacity to cause devastating birth defects. This constant emergence is a stark reminder that we are part of a larger ecosystem, and that the barrier between animal and human pathogens is porous and frequently breached. The threat of bioterrorism looms as well, the deliberate weaponization of the most dangerous of these agents, smallpox, anthrax, plague, turning the tools of nature into instruments of war. But, as the enemy evolves, so do our defenses. Our greatest weapon in this war is knowledge. We have developed an extraordinary toolkit for intelligence gathering, for diagnosing and identifying our unseen enemies. The classic special stains of pathology, the Gram stain for bacteria, the acid-fast stain for mycobacteria, the silver stains that illuminate the delicate structures of fungi, still form the bedrock of diagnosis. But we have moved beyond simply seeing the enemy. Serology allows us to detect the immunological memory of a past or present infection by measuring specific antibodies in the blood. And the molecular revolution has given us our most powerful tools yet. Nucleic acid amplification tests, like PCR, can detect the unique genetic fingerprint of a pathogen with breathtaking speed and sensitivity, allowing for the rapid diagnosis of diseases like tuberculosis or herpes encephalitis from the smallest of samples. And now, next-generation sequencing allows us to read the entire genetic code of all the microbes present in a sample, offering the potential to identify any pathogen, known or unknown, and to track its evolution and the spread of antibiotic resistance in real time. This is the state of the modern war. It is a race between microbial evolution and human ingenuity. The stakes have never been higher. Every day, in laboratories and clinics around the world, pathologists and scientists stand as sentinels on the front lines, using these tools to identify the enemy, understand its strategies, and guide the fight. The war within continues, but we are no longer fighting in the dark.